Welcome to MOOC course on Introduction to Proteogenomics. In the last module, we first studied about genomic revolution, genomic technologies, and in the second module, we have started discussing about proteomics. In my last two lectures, I have tried to give you a very broad overview of proteomics field, different technologies which are being used in my first lecture, and in second lecture, try to focus more on mass spectrometry based proteomics. Today I am going to talk to you about some of our ongoing research work to give you the flavor of how proteomic technologies could be used for different type of applications, especially more focused on clinical proteomics. This in some way is going to summarize that different type of proteomic technologies could be used to address different biological questions. As we go along after this lecture, there will be much more focused discussions about mass spectrometry based proteomics, different softwares and tools involved by various experts of the field who are going to talk about much more detail about how to use various tools for data analysis. So, this is the third lecture of giving you the basics and overview. After this, there will be much more focused discussions and the hands on directly from the workshop. So, just talking about various type of ongoing projects, especially in my laboratory at IIT Bombay, we look at some of the technology platform or the assay development as one of the areas of interest. A major group of the lab works on the infectious disease, especially malaria and dengue, and try to look at different type of either diagnostic biomarkers or looking at the prognosis, how a disease transforms from the non-severe to the severe form and how using proteomics one could try to understand that. Additionally, a major focus in the lab is on the brain tumor, especially cancer biomarkers in collaboration from Patna Memorial Hospital, and then we have various ongoing projects from different collaborators on other model organisms. For this brief presentation, I will focus mainly on two different disease projects, one on the infectious disease and other on the cancer projects, and try to give you a broad understanding about how proteomic technologies could be used to study different type of clinical problems. Let us first talk about infectious disease or malaria proteomics project. Malaria, I am sure all of you are familiar, as the monsoon season starts, people starts getting different type of mosquito bite and get affected from either falciparum malaria, vivax malaria or sometime dengue fever, chikungunya, variety of fever you can term, and it becomes very difficult for the clinicians and the doctors to really make accurate detection of what kind of the organism has caused this particular type of fever. And if you get the right diagnosis, only then you are able to get the right type of treatment, right. So, the interestingly looking at malaria, the trend for malaria pathogens have slightly changed over the time period, especially last couple of years when we have started investigating this problem in India. We found that falciparum malaria have reduced, whereas vivax malaria has quite increased now. And you can see the trends in different regions of India where it shows that now vivax malaria is pretty much on rise. While earlier this particular vivax malaria was thought to be very benign and not causing much of the problems for malaria, but now looks like it is one of the major culprit which is causing a lot of malaria problems in whole India and of course many parts of the world as well. So, however, our major understanding of malaria predominantly depends on the falciparum malaria. A lot of research has happened in that particular pathogen, but when it comes to vivax, it has been very limited because of various reasons of our inability to culture the vivax in the culture system, which is not the case for falciparum, and also the vivax even at a very low parasitemia level, it could cause even the severe effects, which is not the case again for the falciparum. So, current modalities of diagnosis of malaria parasites based on the microscopy, or it could be rapid diagnostic test, or it could be PCR. While microscopy is gold standard, when the pathologist they look at the the parasites in the microscope, and if these are the trained eyes who can look at the parasites well, I think that is very accurate way of detecting parasite. RDTs are very quick, these are based on some antigens when you can do some history based test quickly to tell if a pathogen is coming from the falciparum or vivax or Brix infection, and one could try to get that information, or the PCR could be utilized to be very accurate that which kind of pathogen is causing this particular febrile response. However, each of these methods have their own pros and cons, especially RDTs have lot of false positives. PCR can be done only in the specialized labs, so it needs lot of further testing. And overall limitation for the whole field has been that most of our information is based on falciparum, not vivax. Therefore, everything is we try to put together based on the falciparum and say is that falciparum positive or negative, and accordingly whether this could be vivax. So, there is still need to look at specific antigens for vivax if we really want to investigate this problem. So, in this slide one could look at different type of proteomic approaches, different type of biospecimen to try to investigate this problem. Let us say one question we will try to address: how does the proteome modulates in response to Plasmodium falciparum or vivax infection? To do that, we looked at the patient's blood sample and try to look at the plasma as well as the erythrocytes, the plasma proteome to look at the host responses, and the erythrocyte for looking at the parasite proteins to look at what are the parasite proteins which one could detect directly from the clinical isolates. So, both of these strategies in some way are complementary to give us very vast information about the parasite proteome and the host proteome to find out what is happening in response to this malaria in these patients. So, one of the goal of the project was to look at can we identify some specific biomarkers which could be very unique for the vivax malaria. A lot of work has happened in the lab, but just in a nutshell, in one of the study where over 200 patients were enrolled, we looked at parasite proteome. We also look at which of the parasite proteins are secreted in the plasma, because those are circulating in the blood and those could be much more relevant biomarkers for our study, and therefore, some of these biomarkers we try to now do the bioinformatic analysis and see are they only uniquely present in vivax or whether they are also present in the falciparum. And interestingly, many of at least 5 candidates looks very promising only present in the vivax, which could be next generation diagnostic biomarkers. Now, there is a need to take these leads forward to clone, purify these proteins and then see with the possibilities of having the RDTs based on these protein biomarkers. Additionally, we looked at now a protein microarray based platform to know which antigens are markers of exposure in malaria. To look at this, some of this study can be foundation for doing the population based studies where one could ask various questions. What is the antibody breadth for a population based survey? And in this case, we looked at from the Goa population in collaboration from University of Washington, Professor Pradeep Rathod's lab, variety of patients affected from malaria, both falciparum and vivax were tried to screen, and on the chip we had only the antigens from falciparum or vivax parasites. So, now the patient's serum samples are probed on the chip and we are looking at antibody breadth of the patient suffering from falciparum or malaria. Also, we try to look at the surveillance kind of responses based on the antigens which is shown in this particular the middle segment here, where you can see even based on the age groups one could try to see different type of responses, and in the left side you can see based on the falciparum or vivax the patterns are different for the seroreactive antigens which are recognized by these patients. And finally, if you look at the right panel, we are looking at also the severe and non-severe responses in these patients, and you can see a very different pattern from the non-severe versus the severe patients. So, again this could be one of the interesting approach of looking at the antibody based responses using protein microarrays. Again, to address what are the mechanism of vivax causing the severity, for which we do not have much information, and some of these study I am just talking to you about partially published and some are leads are still unpublished, where goal was to look at if a patient comes to a doctor and they are having the mild fever, whether one could try to look at possibility of them transforming to the severe form and what kind of proteins and metabolites could be indicative of the severity. So, in this slide we did a quantitative mass spectrometry based workflow where the healthy individuals, non-severe vivax malaria patients, severe vivax malaria patients were screened. Along with them we use a reference pool here So, all of this a typical eye tract based mass spectrometry study which we talked in the previous lectures was employed, and what it revealed that there are set of proteins which are very unique to non-severe or the severe YVAX patients, looking at both the proteomics approach, and even after metabolomics also we found that there are some very unique metabolites which are secreted here. Some of the proteins from the acute phase pathway and the cytoskeletal proteins are shown on the screen here. When a protein like serum amyloid A shows from the healthy individuals to the non-severe to the severe patients kind of rise of the change in the abundance of these proteins. Another protein like haptoglobin shows the reduction overall, the concentration is reduced from the non-severe to the severe patients, and that is more logical because the haptoglobin and hemoglobin they form the complex, and therefore you will see the reduction of this particular protein in the severe YVAX patients. Some of the cytoskeletal proteins like titin and vitronectin, they showed the increased response as the disease progresses from the non-severe to the severe type, which is indicative of some of the muscle protein and cytoskeletal protein being secreted from the muscles to the blood stream, and that shows now the patients could be modulated from the non-severe to the severe type. We try to look at various metabolites shown in the right side, and then the red dot shows you the pattern of healthy individual, and this part shows the non-severe and the last part shows the severe patterns. Even by looking at the metabolite profile, one could see at least there are some metabolites, especially various amino acids, they show the change in response to the non-severe and severe type of malaria. So, based on this we are trying to now capture which are all changes one could utilize from the omics technologies, and try to look at what is the mechanistic insight of YVAX malaria severity. We have tried to put together a lot of information from proteomics data and metabolomics data, able not to give you a lot of detail right now for the mechanism, but I am trying to emphasize that how these tools and technology what we are talking can give you the new insights which could probably help you to understand the mechanism of a given disease. So, in this context very first time we are now able to put together various pieces and trying to see that in which way the various changes which are happening which are reflecting from the addition molecule to the inflammation factors, how they are with the change of various type of amino acids and change in the haptoglobin level leading to the oxidative stress or apoptosis, followed by it is influencing the vascular leakage and coagulation pathways, which is eventually causing variety of severe infection which we can see. So, the activation of oxidative stress as well as the the cytoskeletal regulatory molecules could be one of the major mechanism which is contributing toward the severe YVAX malaria. Of course, this is very complex slide which needs lot more time, but I am not going in detail. Here idea is only to give you the the glimpse of what you can try to understand from the proteomics and other omic technologies. Additionally, we are trying to also address more focused questions, like in case of falciparum malaria, there are various type of severe infections happens, like patient could be suffering from the cerebral malaria or renal failure or variety of other complications. All these could be termed as SFM or severe falciparum malaria, and question was could we look at proteomic based alteration in the severe falciparum malaria based complications. In this slide we selected the patient from cerebral malaria, severe anemia, other type of severe falciparum type. When we do these kind of proteomic studies or clinical studies, choosing the right control becomes very crucial and of course challenging as well. So, in this case for the anemic population we choose as a control for severe anemia. We also choose various type of non-severe falciparum patients. We got some meningitis patient for the control as a cerebral malaria. So, you need to have the right type of controls to compare the various type of disease complications. Now, after doing lot of proteomics workflow, this heat map is shown here, which shows that one could actually segregate severe and non-severe falciparum based on some of the proteins shown on the right hand side. And even within each type of severe infection, like you can see cerebral malaria, severe anemia, other type of severe infections, their trends for various proteins are quite different, and therefore this information could be helpful to get a glimpse of what type of severe infections these patients might be undergoing if they are affected from the falciparum malaria. Additionally, we are also interested to look at are there some parasite proteins secreted in the serum or plasma of these individuals, and whether those could be used as the next generation diagnostic biomarkers. because some of the existing biomarker like PFHRP2, while it has been a good lead for the RDTs, but there are many population where now it is being shown that there are some mutations happening and this may not be the best diagnostic biomarker. So, there is definitely need to have the alternative next generation diagnostic biomarkers even for falciparum malaria. And in this light various protein which we try to identify, some of those are already same protein what are available in the existing RDTs. But additionally we are also able to find some new proteins which are secreted from the parasite in the host blood stream or the plasma, and one of those is serine repeat antigen 4 protein, which looks promising again and could be taken forward as a potential biomarker candidate. So, in the nutshell of the malaria project, we now have various leads, various protein targets both from the parasite as well as from the host, and our aim is to look at what happens to the given protein, like let us say alpha 1 anti-chymotrypsin or alpha 2 HS glycoprotein or complement C3 across different type of complication, from the non-severe falciparum to the non-severe vivax to the dengue fever and severe type of falciparum and vivax. So, what happened to the same protein across multiple infection, and if there is a real signal coming out of in a specific type of infection, you will see a different trend. Now, some of this information we are trying to take forward to develop some of the the possible kits or the assays for the better diagnosis and prognosis of the patients. So, in general the conclusion for the first part of malaria proteomics is we are looking at various approaches of serum proteomics and metabolomic analysis to look for the prognostic biomarkers and understand the host responses. We are also looking at various type of parasite proteins in the serum to look at the potential biomarkers for the diagnosis. We have also investigated the non-severe to severe comparison to try to look at the severity of different complications both in the falciparum and vivax, and actually nothing was available information for the vivax, and we have also done much more in depth investigation of the different type of severe falciparum complications. And an offshoot of the overall project is also looking at the antibody breadth and looking at humoral responses of using the protein microarray based workflow to look at various type of markers of exposure of malaria. So, this is where you can see to investigate one clinical problem you can utilize different type of technologies from gel based to mass spectrometry to microarrays and to SPR even to test out the protein drug interactions to then try to understand comprehensively a given problem or a given system. So, let us now shift gear from infectious disease, moving on to one of the clinical problem of a great relevance of the cancer, especially the brain tumors, which is very deadly tumor which affects lot of patients who do not survive after very long if they are diagnosed with the brain tumors. Depending on the location of the in which region the tumor is, the brain tumor could be termed as the gliomas if they are deriving from the glial cell, or meningioma if they derive from the meninges, or medulloblastoma if they are in the pediatric tumors. So, to do this kind of project we collaborated from doctors from Tata Memorial Hospital as well as ACTRAC in Mumbai, and we try to utilize various type of workflows and approaches how to use proteomics to investigate these problems. But let us kind of give you a brief of gliomas and meningiomas, which are very challenging brain tumors. Most of the these brain tumors are very heterogeneous, as well as if you look at the available information from WHO, that is more based on the cell morphology and some of the immunohistochemistry based biomarkers. There has been extensive genomics which has happened in this especially the glioblastoma multiforme brain tumor, but not much of the overall proteomics or the proteogenomics investigation has happened so far for various type of the low grade gliomas and meningiomas and other type of brain tumors. So, on one hand we definitely need good repositories or for the biospecimen to do these kind of research. Additionally, there are a lot of challenges of the heterogeneity of these tumors which we have described in some of these review articles, which show the challenges of doing this kind of research, because you need large number of samples, and even if you have large number of samples, the patients will be affected from the variety of issues, and therefore so much heterogeneity is there even from the same patient. So, to investigate that you need of course complementary approaches, you need large number of samples, you need very robust data analysis workflow, and together only you can try to obtain some information. So, let us say how we can use the proteomics workflow to address some of these problems. So, you can get variety of sample type either from tissue, serum or the cerebrospinal fluid. Now use this sample type biological specimen to do discovery either using mass spectrometry based workflow or protein microarray based workflow, and after identifying the potential candidate targets, then one could go to validate the biomarker using targeted proteomics based workflow. So, some of the technology which we talked in my first lecture, let us try to see how we can put them together and use them in this kind of clinical problem. So, we wanted to ask a question whether this kind of proteomics investigation which we are trying to address for a brain tumor could identify some of the key networks and potential targets for different grades of meningioma brain tumors. So, we are now looking at meningioma, and then we are using 3 different type of complementary approaches. One is global proteomics, where aim is to look at all possible proteins and use their quantitation using either iTrack or TMT based workflow, or use the global proteomics workflow using the label free analysis, which is LFQ, label free quantification, or DIA, data independent acquisition, or we do the phosphoproteome analysis just to look at the enriched phosphopeptide residues available from these peptides. To do this, let us say from the same patients, the brain tumor sample when it comes, you can do the protein extraction and split those particular peptides after digestion into multiple tubes, and now each tube containing peptides could be utilized for either iTrack or TMT workflow or LFQ or DIA workflow, or you can use for eventually for the validation strategy using MRM or SRM assays, or do some multiplex assays in future. So, now the same sample you prepare in sufficient amount which could be utilized for different type of strategies. So, let us go one by one. We took the patients of meningioma, different grades of meningioma patients, especially grade 1, 2 and 3. 3 are very few. So, we had mainly grade 1 and grade 2. Grade 2 there are more malignant patients. We used the iTrack based work strategy as well as TMT based workflows to try to compare different grades of these patients. Reliably we could get almost 3000 proteins from the quantitative comparisons, and then we also use the same samples to now look at label free quantification or data independent acquisition. Using DIA workflow, we could actually reach out now to almost more than 4000 proteins, which is after much stringent screening that we can screen so many patients, so many proteins from each patient, which now we can quantify in the label free manner. So, same patient samples, you are now trying to use either label based strategy for doing iTrack based quantification, or you are using the label free quantification to compare how the controls look different from the grade 1 patient or the grade 2 patient. Similarly, we try to enrich the phosphopeptides after passing through titanium dioxide column, and then use this phospho enriched fraction for the analysis, and now we try to compare all the 3 information from the label free, iTrack and phosphoproteome. While each of these provides some set of unique information, but what we are also curious to see which fraction is actually showing common pattern, because that shows that these are reproducible from different independent technologies, as well as these are also showing us the trends for the phosphopeptides or the PTA modification. So, almost 208 proteins or a sizable amount of stringent proteins showed a common pattern emerging from various independent technologies. Some of those we took forward, and now we try to look at the plotting in the heat map formation, how that compares from the meningioma grade 1 and grade 2. And as you can see the green line here, this part is for the meningioma grade 2, which looks quite homogeneous, whereas the red line shows the heat map for the meningioma grade 1 patients, and this shows that there is lot of heterogeneity even in the grade 1 patient. Not all grade patients look exactly same, a set of patients look slightly different than the other set of patients. When we try to plot the PCA plots to look at the pattern based on these proteins, can we now segregate the patient population? It looks interesting that these are the 2 type of controls, dura mater and arachnoid. They are actually different from the different origin of the brain, so they look different here. The grade 1 patient look like they are forming 2 subtypes, and then grade 2 they are quite homogeneous, they look very much together. And based on these information, we are now finding some interesting protein leads, some kinases which could be taken forward. But what is most interesting from these kind of proteomics and big omics studies, you get to see large number of changes, and you try to put them together and analyze based on the pathway analysis that most of the changes are perturbing which type of network and pathways. So, looking at this information, we found that integrin pathway as well as the PI3 AKT pathway were greatly affected because of the meningioma disease. So, now the logical flow was to try to investigate this problem and look at this pathway in much more detail. Many of the the network, this is what is shown here, these proteins were identified from our discovery workflows. So, our question was whether integrin and PI3 AKT pathway have some concerted effect on the meningioma pathobiology. To do this, in the collaboration from Dr. Nell's lab from Imperial College London, one of my students, she went and looked at the patient derived cell line of meningioma and then treated them with one of the inhibitor, which is ILK integrin linked kinases, to look at what is the effect of this particular inhibitor on these meningioma patient cell lines, because our aim was to look at some of the targets of the PI3 AKT pathway and whether this inhibitor could actually block or affect some of these proteins. And interestingly, it showed that this inhibitor had the effect where it could actually potentially affect the target which we have talked in the network analysis. And then after doing the real time PCR analysis, some of these targets were again confirmed that this particular inhibitor has perturbed these genes of interest. So, this study is still undergoing and currently underway to do more of the biological replicates, but the promise here is that if you can get these kind of therapies, this could be a surrogate way of adjuvant therapy, where if you can't treat the patients with the surgery, can you use some inhibitors to at least try to control the tumor for some time. Additionally, the clinicians gave us some sort of focus questions to address, especially based on the radiological observation, whether the skull based or supratentorial locations of the brain may have an effect at the proteome level. To do this, now if you have done the proteomics using label free, you can now use the same data and now just try to analyze that in different manners. So, in this case now we try to look at how the skull based or supratentorial brain tumors are actually getting segregated in the meningioma population. And very interestingly, it looks like they are very clear segregation of these two type of subtypes based on the radiological observation. So, looks like radiology is much more closer to the molecular signatures as compared to pathological observations. But of course, some of these we need to still take forward with more validation. But what is coming out interestingly, after looking at these data and doing big data analysis from the artificial neural network, we could now see the impact of some of the positive regulators and negative regulators and how they are going to affect these type of brain tumors. So, this is where you can see that how you can start from a clinical problem of interest, identify the targets and then do big data analysis to try to get some sort of meaningful conclusion out of this information. Additionally, rather than using mass spectrometry alone, we also try to utilize the complementary technology of protein microarrays to address the question, can we identify autoantibody signatures in meningioma patients. And goal here was to use protein arrays platform, take patients clinical sample, serum sample, probe them on the chip, and if there is any antibody generated in the patient sample, they will bind on the chip which is having all the protein antigens printed. To do this work, we did collaboration from Johns Hopkins, where we have all these almost 19,000 proteins printed on the chip. Now, if we see a signal from the autoantibody, which is shown in the next slide, this looks like these are the control samples, healthy individual, meningioma grade 1, meningioma grade 2 patients, and some proteins are showing very strong response, and these could be potential autoantibody biomarkers for the detection of this particular tumor. So, coming back to the various workflows which one could utilize, we have used both mass spectrometry and protein microarray based workflow. Then there is a need to do validation, and for validation you should not limit yourself to any specific technology, rather use whatever is available to you, ELISA, Western blot or microarrays, or even looking at the target based workflows to get more confidence. And many times you will see that while antibodies are not able to detect and make a lot of changes because they might have been raised for a given epitope, but your other type of complementary technologies are showing you much more higher changes, and therefore then you can gain more confidence by having multiple technologies to validate the proteins of interest. We have also tried to address specific questions from the clinicians, especially in the glioblastoma multiforme patient, looking at the location of the brain tumors, what could be the impact of the tumor location to the ventricle region, is it very close or is it far off. These patients will be surviving less, these will survive longer. It is much more aggressive tumors here, known as SVD positive or SVD negative, and can we look at the proteomic signature to try to classify these kind of tumors. In another project in collaboration from Professor Gilead's lab in Tel Aviv University in Israel, we tried to investigate the colorectal cancer problem, colon adenocarcinomas, and we found the panel of 9 proteins looked very reproducible across large number of patients affected from the colon cancer in the Middle East countries, and we are now trying to validate this in also Indian patient samples. Another interesting problem is medulloblastoma, the pediatric brain tumors. Again, we are trying to provide the proteomic based subtype signatures from the various type of the patients affected from either the Wnt type or the SHH type or group 3 and group 4, what could be the possible protein alteration in these type of the children, this pediatric brain tumor population. Finally, we are trying to look at variety of approaches of proteomics. Also, we are trying to use the various basic science tools of doing the transfection and understanding the effect of these mutations on these particular tumor type. We are developing various type of clinical assays in collaboration from the industrial partner of targeted based proteomics. So, overall I try to give you the glimpse of how to put together proteomic technologies in context of various clinical problem. We try to address 2 clinical problems of looking at the severity of infection in the case of malaria, or also looking at what could be the possible cues and subtype molecular classification of brain tumors. So, eventually these kind of technologies may provide you and give you the possible targets which could be translational potential to take the leads from the bench side to the bedside, and that is what is the goal for many of our labs working in the areas of genomics and proteomics. But how now to leverage this information and integrate that as a part of proteogenomics, this is what this workshop and course is about. We would like to utilize the proteogenomic information for the better patient care. Thank you.